I think we're ready to get started with our session one. So I'm very happy to be here. I'm Scott Tykodi from Seattle, University of Washington, and Fred Hutch campus. And for this session, joined by my colleague, Pavlos Msaouel, to run the session. So I invite all of the speakers to come up. We have 4 chairs for our 4 speakers for this session. So our first scientific session, short talks, but plenty of time to ask questions. So think of your questions. We're going to take questions for each speaker after they finish their presentation individually, not wait till the end. Sampling from all of the wonderful research supported by the DoD Kidney Cancer Research Program for this session. There isn't time in this program to have every funded speaker give a talk, and so we try to have a broad theme. And as a physician that takes care of kidney cancer patients, we're always looking to see what's in development, what's coming, what might be a target, a vulnerability in cancer that can be exploited for therapy. And so that was the overall theme for the speakers in this session. So thank you, and let me introduce Dr. Msaouel to make a few comments.
Thank you, Scott. It's a really pleasure to chair this session. It was really fun for us to select the speakers, focusing on novel MOAs, which are needed both for clear cell kidney cancer and other histologies. So we're very excited to be here. And I would like to invite to talk Dr. Jiang first about her research on new potential modalities for kidney cancer. She is from the University of Texas Health San Antonio.
Yeah, thank you for the kind introduction and invitation. We're developing the potential therapeutics for the treatment of the metastatic clear cell renal cell carcinoma, also called KCRCC. As we know, there are up to 65,000 patients diagnosed each year in the US with KCRCC. And then about 24,000 patients are present with stage 4 metastatic KCRCC. And then after reaching to the stage 4, the survival is not good. It's only 12 to 13% chance of surviving in 5 years, and also associated with a huge medical costs, about $2.5 billion each year. So we're mainly focusing on the adenosine A2 receptor. We believe this is a potential drug therapeutic target. The mechanism of action is normally for the cancer cells as well as non-cancer cells, they can release the ATP. The ATP outside the cell is very unstable. They can be degraded into the adenosine. Then adenosine can target it to the adenosine receptors. There are total 4 types of adenosine receptors. We're particularly interested in the A2A and A2B receptors. The main reason is this receptor, like A2A, this activation is immune suppressive. So they can suppress the activation of the T cells. Then because it's suppressing of the immune system, then can preventing the immune cell to target on the cancer. Also, we focus on the A2B receptor. The A2B has the 2 mechanisms of actions. One mechanism is the A2B activation can suppressing the other type of the associated immune cell, like antigen-presenting cells. Because of suppression of antigen-presenting cells, they also can prevent activation of the immune system. Another mechanism of action for A2B is they can promoting the cancer growth and also the migration, which are associated with the tumor metastases. So we'll try to develop the inhibitors that can blocking these two receptors, and then they can suppressing these 3 associated pathways. So we identified the new compound, and this new compound as a dual A2A and A2B inverse agonist. Initially, we thought this compound is an antagonist, but then through the more thorough studies, we found that this compound actually can fit into the agonist pocket. However, it's surprising the mechanism is a function as an inverse agonist. The difference between antagonist and the inverse agonist is for antagonists, they only function in the presence of antagonist. In this case, it's adenosine. However, for the inverse agonist, they function not only in the presence of the antagonist, agonist, but also they can inhibit the basal level of the activation. So it's showing here. Also, we're looking at the cancer migration assay. We found if we treat it with the compound, they can inhibit not only with the presence of adenosine, but also inhibit the basal level of the migration. You can see compared to the basal level, it's inhibited even more. So then we did further studies. One is we're looking at the triple-negative breast cancer. We found that if we treatment with this compound, and then they can not only inhibit block the primary tumors, but also improve the lifespan and also have the dramatic effect on the metastatic cancer, especially metastasized into the bone. Also, they can eliminate or reduce circulating cancer, both in the blood as also in the bone marrow. Also, we further characterized the drug characteristics. We found that they have favorable drug features. They have low toxicity in the multiple organs we tested. Also, no effect on the liver enzymes level. Also, this compound cannot penetrate through the blood-brain barrier. And they also have a low off-target effect. Also, we did looking at the overall health in the animal studies. It looks good. Also, the drug we did DMPK. We found that the drug is relatively stable. Also, have the good accumulation in the tumor. So the reason we're focusing on the KCRC is because this in the KCRC in the database it shows this A2B receptor is highly activated, increase the expression in the KCRC. Also, the expression of the A2B receptor is directly correlated with cancer prognosis. So you have a higher expression of A2R, A2BR, and then they have a poor prognosis. That means they have associated with a shorter lifespan. So then for the, right now we're doing more to try to understand the underlying mechanism. One question we're asking is whether this inverse agonist compound can really inhibit the immune suppressive tumor microenvironment in the KCRC. What is the major pathways involved? Also, we're really, most importantly, is to developing this new cancer therapeutics for treating the metastatic KCRC. Right now, we have several ideal candidates, but now we try to identify the lead compound. And also, if necessary, we'll try to optimize the lead compound. And this study is currently supported by the DoD grant. Last but not least, we really like to acknowledge the people involved in the work, the people in my lab, researchers, also the collaboration with the Riverwalk Therapeutics, also our advisors. They provide very valuable feedbacks. I especially want to thank the great support from KidneyCAN. Thank you very much.
Thank you for sharing this exciting talk. There's definitely a lot of interest in harnessing the adenosine pathway and how it can modulate the immune microenvironment. If there are any questions, from Nizar.
So do you think this blockade of the dual A2A and B by itself as a single agent has activity in clear cell RCC, or do you think this is best in combinations? Have you done any clinical work to see as a single agent it has activity?
Yes, so this is a single compound. They have a dual activities. They can block in both A2A and A2B. They both on the high affinity. Especially for A2B, the affinity is quite high. It's in the single nanomolar range. In the A2A, it's in the double-digit nanomolar range.
Yeah, I understand, but do you have any clinical data in patients that as a single agent, this drug, this compound, has activity in clear cell RCC?
Right now, we're doing the preclinical study, so it's not reaching to the clinical phase. That's our goal. Yeah.
Speaking from experience, Nizara, that's why you're focusing on that. Another question?
Hi. So this concept of inverse agonist is very intriguing to me. I don't really know much about it. I'm just curious, one of the potential issues with the current antagonists is that they have to outcompete adenosine in the tumor microenvironment. You mentioned that the inverse agonist works in the absence of adenosine, but what is the potency when there's a lot of adenosine around?
Right. So adenosine is typically is a low binding, is a low affinity, especially for A2B, and then for the compound is a high affinity because it's a magnitude greater the affinity. And so for we compared with the current antagonist for the adenosine antagonist A2A and A2B, and currently there is one program ongoing. And so that one already reaching the phase 2 clinical trial on the different cancers. So we compare with the breast cancer, our inverse agonist, the potency is greater. And also we applied less frequencies and much lower doses.
Thank you.
One more question, one last question.
How ubiquitous is A2B receptor expression in the microenvironment compared to A2A? So I'm thinking back to like Phil Rappold's paper in Cancer Discovery from Dr. Hakimi's group that they showed that that myeloid inflammatory state is associated with metastases, and using like a ciforadenant-like compound reduces metastases in a P53 knockout mouse. What do you expect the impact to be with the A2B inverse agonism in that context?
For A2B, it's they not only on the immune cells like APC cells, they also on the cancer. But for A2A, there is no such correlation. So we feel like another unique part is targeting the A2B. You basically have a double benefits. Right.
Thank you.
Thank you.
In the interest of time, we'll leave hopefully time for questions in the end. I would like to invite Dr. Lisa Henske, who is a physician scientist and a kindred spirit at Dana-Farber, who is going to give a very exciting talk about a tremendous unmet need, the need for novel therapeutic targets for chromophobe kidney cancer.
Thank you. I am super excited to be here, and hopefully you'll be very excited about chromophobe by the time I finish. I'm going to start by acknowledging the amazing people I work with, including Tony, Vincent Zhu, David Braun, Carmen Priolo, who is my partner in everything that we do in chromophobe, Bill Sellers, I'll be telling you a bit about our work with him, and importantly, the amazing people in my group, including 5 who are here today, all working on chromophobe. So I'm going to talk about 3 things. I'm going to be sharing mostly unpublished data that reflect the directions that we are taking right now, and I really hope that you'll give us some idea. So the first is ferroptosis, the second is a new project targeting the c-KIT, tyrosine kinase, and the last is a new mouse model we're excited about. So just to start, why are chromophobe tumors so interesting and unusual? Well, they arise from this very specific cell in the distal nephron called the intercalated cell. And the intercalated cell has this incredibly difficult job. It maintains acid-base homeostasis in the body by secreting hydrogen ions into the urinary space. And because of this, these intercalated cells are filled with mitochondria. And we think that's part of the problem with chromophobe, that these mitochondria-rich cells develop mutations or mitochondrial dysmorphogenesis, I guess, leading to increased ROS stress. And we know that there are very few driver mutations in chromophobe. About maybe 30% have mutations somewhere in the PTEN pathway, another 30% P53 mutations, but most of them have no identified driver mutations, yet they have the highest incidence of ND4 and ND5 mitochondrial gene mutations in the TCGA. So we and another group found that glutathione levels are really, really high in chromophobe. It's kind of amazing, 50 to 100 times higher than normal kidney. And so you might have thought that this would make these tumor cells very resistant to oxidative stress. But in fact, the opposite is true. These tumors are very sensitive to oxidative stress, more sensitive, for example, than clear cell cancer. And ROS stress leads to ferroptotic cell death in these tumors. So what is ferroptosis? Ferroptosis is an iron-catalyzed reactive oxygen species-induced form of cell death that's distinct from apoptosis. There are sort of two ways that tumors and cells can resist oxidative stress. One is glutathione peroxidase and glutathione itself, and the other, which you'll be hearing about tomorrow from Samer Salem in the lab, involves a separate pathway involving the ferroptosis suppressor protein. So we think that one hypothesis, with totally unproven, is that these tumor cells usually, when they try to enter the bloodstream, undergo ferroptotic cell death, because it's known that once a tumor cell reaches the blood, it's exposed to high levels of oxygen and iron. And what we're wondering is if the metastatic cells may actually be the ones that can resist this early ferroptotic cell death. And it's interesting that P53 mutations and sarcomatoid morphology are both associated with metastatic potential in chromophobe. So in work that Joelle Chamy was doing, she's here today, we're trying to understand whether this is the case, whether tumor cells develop resistance through P53, allowing them to metastasize. And this is some unpublished work from Joelle. We've developed some tumor cells in cell culture that over the course of about 2 months have developed resistance to one of the main ferroptosis-inducing drugs, which is called IKE. You can see the cells here, and Joelle is just in the process of analyzing them. Not surprisingly, these cells have upregulated the SLC7A11, the transporter that is the target of IKE, but they also seem to be resistant to other forms of cell death, and we have some RNA sequencing that should be coming back next week to tell us more about what's going on with these cells. We think that this might also be the reason why it's so hard to establish chromophobe cultures. We have so few tools to study chromophobe, and we're wondering if what's happening is you put these tumors into cell culture, they quickly undergo ferroptosis. So I would beg you to buy some NAC, it's cheap, and next time a chromophobe comes through your lab to try culturing the cells in the presence of NAC, perhaps this would help them overcome this ferroptotic checkpoint and establish themselves in cell culture. Next, I want to talk about KIT. So KIT, as you know, is a tyrosine kinase receptor. Activating mutations are found in GI stromal tumors and other tumor types. And of course, we have very effective inhibitors like imatinib and dasatinib. But what's happening in chromophobe is different. So chromophobes have, and I just would mention that KIT is the marker of intercalated cell. If you're a nephrologist or a scientist studying intercalated cells, you would pull down intercalated cells from the kidney using c-KIT antibodies. So c-KIT is highly expressed in chromophobe. That's shown here. It has the highest expression of any tumor type in the TCGA, way higher than clear cell, many-fold higher in chromophobes than in normal kidney. And it is expressed in metastatic renal cell carcinoma. That's shown here, expression in both a primary and a metastatic tumor. And in work that Michel El-Shaweri has done, he's also here today, and Hadi Mansour, we see that KIT is clearly expressed in the cell lines that we have available. And in very early data, again, unpublished, we are seeing a dose response in one of the kidney cancer, chromophobe kidney cancer models called UOK276, developed by Marston Linehan. And here in blue, you're seeing the clear cell 7860 cells have no response to KIT antibody-drug conjugates, but the two of the cell lines do have a clear response. So we're hopeful that this will be a new avenue to treatment. And then lastly, I want to tell you about a new model of chromophobe that we're excited about. So this is a inducible model in which TSC2 is deleted at E17. And what's interesting is that some of the tumors that you can see here in the mouse really resemble the chromophobe tumors in humans. And I just would mention again that mutations in the PTEN, mTOR, TSC pathway are found in a subset of sporadic chromophobes. So we did single-cell RNA sequencing on the kidneys from these mice, and we found a distinct subset of tumor cells that express KIT, so we're excited about it from the perspective of the KIT project, and also that express the hallmark genes of chromophobe that were identified by Lindgren et al. Back in 2017. So we're wondering whether this model might be a genetic model that could be useful in studying chromophobe. So I'll end there. I've told you about where we're headed with ferroptosis. You'll hear more about that from Samer tomorrow. The very new concept that maybe c-KIT antibody-drug conjugates could be useful in chromophobe. And lastly, a new model with chromophobe-like c-KIT positive tumor cells that's based on TSC2 inactivation. And lastly, I would say it really takes a village to think about tackling chromophobe, or maybe as Brian Lewis said, it takes a really great team. And I want to thank so many of you in the audience who have really gone above and beyond to help us think about chromophobe and help move things forward. And I think we have the village now. We're here today. I think progress for chromophobe is possible. So thank you very much.
Got to get better at this. Lisa, a quick question. The cells you've developed that are now resistant to ferroptosis inducers, and I know you're just analyzing them now, but our lab and Kivach Biersoy's lab has shown that lipoprotein uptake can be particularly good for ferroptosis resistance because they carry vitamin E, vitamin D, and this is true for high-density lipoproteins, low-density lipoproteins. And some of there's receptors for this like SCARB1, et cetera, or glycosaminoglycans can really help in this process. Do you guys see any evidence of those genes going up?
Oh, that's so interesting, Celeste. Well, hopefully Joelle will have the data back next week and we can start looking for that. That's very interesting. Thank you.
I think I got the thing. I'm over here. Do you think c-KIT signaling is involved, or is it just a good marker that you're going to use for something to...
Yeah, we're trying to understand whether the signaling is important. The initial studies that we did with imatinib and dasatinib, we didn't see very much effect, but most recently Hadi, who's here today, has been stimulating the cells with stem cell factor, and we do see some signaling, so I think it's still unclear. It certainly would be convenient if it's the signaling, because then we would have the drugs already available.
Lisa, over here. Great talk, as always. I can vouch for that. Developing a chromophobe cell line from patient-derived cell line it's very tough. With regards to ferroptosis and c-KIT, you don't think there is a crosstalk? c-KIT somehow is involved in regulating?
Yeah, we haven't thought about that, but that's a great suggestion. Maybe there is some kind of crosstalk between KIT. I don't think anybody knows why KIT would be so prominently expressed specifically on intercalated cells. So presumably it has some function for those cells.
And through your RNA-seq, if I'm not wrong, you identified a lot of VATPase
Yes,
genes involved,
yeah.
but nothing on the plasma membrane proton pumps.
Well, the plasma membrane of the intercalated cell has these proton pumps, the VATPase, that would otherwise be located, of course, on the lysosomal surface involved in acidification of the lysosome. Intercalated cells have that very same VATPase on their plasma membrane.
Nice talk. At least from a pathologist perspective, I feel I mean, it's a challenging problem, obviously, and I feel like having a canonical TSC mutation in a kidney tumor in a cell line model or animal model, I mean, I think might push down the tumor around the mTOR TSC pathway, which are possibly now distinct neoplasms. So I think seeing the canonical copy number changes of chromophobe RCC, I mean, that's another suggestion, just to check, like, are we seeing those in these tumors? And even there can be some morphologic overlap in between those two. So I feel, because now we know that less than 5% of the overall RCC are mTOR TSC driven, and they're canonical, and they can have some morphologic overlap. So it's good to keep something on the radar.
Yeah, I think this whole issue of the chromosomal losses in chromophobe is so hard to think about. And those, yes, as you know, those do not occur in the chromophobes that occur in Birt-Hogdebay syndrome and tuberous sclerosis. So, and why chromophobes would have those chromosomal losses is completely mysterious. But I would say that, at least maybe you can tell me whether this is true. It seems that in the metastatic tumors, in most cases, they have somehow overcome that chromosomal loss, and they have now 2 copies of those chromosomes.
Yes, I think in the metastatic state, I think I absolutely agree. I think the CNV pattern does get more complex and complicated, including some chromosomal gains. But yeah, I think this CNV problem is a challenge.
Thank you.
Last question, please. Yeah, very nice talk. I'm really curious about the high glutathione, and that didn't actually provide protection. So presumably it's an adaptive response to this underlying ROS stress. So what are the vulnerabilities that you'd get just from the synthesis of glutathione? You know, talk about XCT, but other things like NRF2 pathways, et cetera.
Yeah, so basically I do think that what might be happening is that these mitochondrial rich cells have sort of a vicious cycle where they get more ROS either through mutations or some other mechanism, then the mitochondria are even more damaged and they make more and more ROS. And yes, that should cause many different metabolic vulnerabilities in addition to the cysteine uptake one, which we've focused on right now, because the synthesis of glutathione is very expensive for those cells. So that could lead to other targetable, targetable pathways. I totally agree. NRF2 and other mechanisms in there.
Thank you.
Thank you so much.
Very good. Let me invite our third speaker of the session up to the podium, Dr. Abhishek Chakraborty from Cleveland Clinic Foundation.
Thank you. So as you all heard, all of these are coming from grants that were funded by the DoD. So I kept my title also exactly what was the title of the grant, essentially, because you'll see a lot of this is work in progress, but there's some background over here about why we got interested in this particular protein called BCL-XL. I don't have any disclosures. I have some personal stock irrelevant to this talk. BCL-XL as a targetable vulnerability basically came out from some work that I started doing as a postdoctoral fellow, and we found that BCL-XL loss was preferentially leading to cell death in the kidney lineage cells. Initially, we thought that this was just going to be just an anti-apoptotic protein that was going to be pan-essential. We started working on this just after I started my lab, and soon after we realized that there was some specific magic built into the dependency. In particular, all kidney cancer cells were not susceptible to inactivation of BCL-XL. Only about 40% were. And if we looked at what were the drivers, or at least what are the biomarkers initially for BCL-XL dependent versus independent cell lines, we found that there were some clear hallmarks that were up in the sensitive cells. One was epithelial mesenchymal transition, the almost ubiquitous p53 pathway, and then also cytokine interferon responses, all of which were already high under basal conditions, but even further triggered up when we inactivated BCL-XL in these cells. This was one really remarkable picture because epithelial mesenchymal transition really from a clinical standpoint is really interesting, associated with very aggressive tumor features, therapeutic resistance, and so we started thinking more about whether this could be a driver of BCL-XL dependence. As you can see, the CD44 marker, which is a marker for mesenchymal or stem-like cells, much higher in the BCL-XL sensitive cell lines or the BCL-XL dependent cell lines than the independent cell lines. But moreover, we could take a BCL-XL inhibitor insensitive cell line, we could treat it with a molecule like TGF-beta, which promotes epithelial to mesenchymal transition, and turning the cells mesenchymal make them more sensitive to the BCL-XL inhibitor. So this was actually a causal event. So this was really remarkable. We just had never thought of cell state and apoptosis in this way, at least in the past in the context of kidney cancer. But soon we realized that we were not alone. There were others who had actually made this link with cell state and BCL-XL dependence. One story came from Ben Stanger's group, where essentially they had identified that BCL-XL was a really critical anti-apoptotic protein, especially in cells that were nutrient deprived, that were essentially in a dormant state, almost resembling a stem-like state. But more directly, Chris Wood's group at Duke had essentially found in other lineages outside of kidney cancer that mesenchymal state was almost a direct biomarker for BCL-XL dependence. You could turn an epithelial cell into a more mesenchymal cell, and now it became more dependent on BCL-XL, so almost like dovetailing exactly with the data that we were having in kidney cancer. So this became our overarching hypothesis that BCL-XL was a critical vulnerability and especially would be relevant in the more aggressive chemo and immunotherapy resistant tumors. And this is what we need to be pursuing over here. So before Tony gets really mad at me, I'm going to tell you why this is clinically important. The first and foremost question we became interested in, how would this be applicable in the clinic? We are finding about 40% of the renal cell lines are being sensitive to this drug, but what would that mean in the context of the tumor samples? How many patients would we imagine would be benefiting from this? And then the other question became relevant in the context of the mechanisms over here. The cytokine storm that we are seeing in response to the BCL-XL inhibitor, what does that mean for immunological responses? What are the mechanisms of this cell state conversion and BCL-XL dependence over here? So these really became the key questions that we began answering in our particular proposal. So what we have done right now is we have generated a panel of gene expression data points essentially that are allowing us to develop a training model at least that is predictive of BCL-XL dependence. It's not perfect as yet. The R values are great, but occasionally we find a dependent cell line score on one extreme versus the other. Largely a pretty good straight line that we are seeing over here. We are right now working with Dr. Chris Sarosiak's lab at Harvard School of Public Health. We are taking human RCC samples and then we are trying, he does the BH3 profiling in his lab. We are using that as an assay alongside the BCL-XL inhibitors to try and annotate tumors as being sensitive or insensitive to the inhibitor, and then using transcriptomic signatures to identify what are going to be the key drivers that will be associated with BCL-XL response in these particular cells. The second question that we are asking is how would this chemokine or cytokine storm that we see in the, upon treatment with the BCL-XL inhibitor regulating immune cell responses, and in particular, given the interferon responses and the interleukins that are being triggered on as a result of BCL-XL blockade, we have started looking at the NK cell responses and T cell responses in this context. One very preliminary data set that I can share with you all is even under concentrations that are sublethal with the BCL-XL inhibitor, just treating RCC cell lines with this inhibitor sensitizes them to NK cell mediated killing. It's a pretty remarkable result. Honestly, we were surprised at how well this worked even the first time we tried it. So this could be something to think about in the context of everybody who's now thinking about NK cell-based therapies. Could we tag along a low-dose BCL-XL inhibitor in order to sensitize the tumors to response over here? And we're doing the same thing right now with the T cell responses as well. Finally, we are testing mechanistically why these cell state changes might be driving BCL-XL dependence. To do this, what we have right now done is we have taken, remember I told you all that you can treat the cells with TGF-beta, you can make them more mesenchymal, and now they become more sensitive to the BCL-XL inhibitor. So we have done a transcriptomic profiling, we have identified the massive changes that occur in these cells, at the transcriptional level when we trigger these cell state changes. We have taken and made curated CRISPR libraries of all of these genes that are differentially regulated, and we are now screening them to identify what are the key changes that are driving dependence to BCL-XL inhibitors. So hopefully we'll be able to find both chemosensitizers but also resistant mechanisms through these types of CRISPR screens. So to summarize very quickly, in a normal cell, Whenever a normal epithelium sheds cells, this happens all the time. It's a process called anoikis that is triggered on. The shed cells are immediately, they turn on signals that promote them to die. But in a tumor cell, these cells live despite they're coming off of this epithelial lining. And this is what we think is happening with the more mesenchymal cells. They're coming off of their epithelial state. But if anything now, instead of dying, they're surviving, but exclusively because of their dependence on BCL-XL activity. You block BCL-XL, you not only kill these cells, but also trigger on these chemokine, cytokine storms, which are now allowing you to have secondary and tertiary vulnerabilities that we can exploit. We are also seeing many metabolic vulnerabilities, which I did not have time to talk about today, and we are doing synergy analysis. One example is the mTOR inhibitor, but then there are a number of other inhibitors that we are testing alongside our BCL-XL inhibitor to identify ways by which we can synergize these two drug-drug interaction modalities. And then also trying to identify, just using general CRISPR screens, ways by which we can take, the 60% cells that are not sensitive to BCL-XL, how can we target those cells? What are the key reasons why they are resisting BCL-XL inhibitor response? So I'll stop there. I really want to thank the organizing committee and for all of you all for listening to this presentation. The work initially was started by funding from Cleveland Clinic and Case Western through the seed money to my lab, but subsequently has been supported by a KCRP Idea Award. The work was led by two people in my lab, primarily Smriti Maganti, who's a research tech, and then Treg Grubb, who recently graduated and is now a postdoctoral fellow in my lab, Chris Sarosiak, and then all the other folks in Bill Kalin's lab at the Broad Institute and the Cleveland Clinic, who have been really instrumental in supporting all of our work. I'll take any questions. Thank you. I'm going to skip this one. So,
Quick question here.
Sorry, yeah.
Okay. That's me. Okay. So a great question. Great presentation. Couple quick questions. One is that you showed that BCL-XL blockades showing responsive in 40% of those cell lines in RCC. But those RCC showing mesenchymal phenotype compared with other ones who don't respond?
Yes. So we basically annotated them almost for everything that you could think about: P53 status, VHL, HIF status, whatever, HIF1, HIF2, et cetera, Of all the annotations, the only thing that eventually panned out was their mesenchymal signature. That CD44 expression levels became the key biomarker that allowed us to distinguish between the BCL-XL sensitive versus the insensitive.
All right. Yeah. The second question: what's the rationale behind BCL blockades with the NK cell responsiveness? I'm not sure I understand that.
So if you treat the cells with the BCL-XL inhibitor, you get a significant interferon response, but also several interleukins the expression of several interleukins is going up. And the goal is that essentially these cells are now going to secrete out chemokines that are going to become almost chemoattractants for the immune cells. So the NK cells became one of the proxies for that particular idea. Yeah.
May I ask you a very naive question? My understanding is that the BCL-XL inhibitors are not very selective. So have you looked at all your immunological impacts in normal renal cells?
Yeah, so that was true maybe 10 years ago, I would say. So the earlier inhibitors that were available were targeting many of the other BCL family members also alongside BCL-XL. The more recent inhibitors, especially the one that we are using made by AbbVie, is a very highly selective BCL-XL inhibitors. It is an inhibitor, but there is still a bit of a problem in terms of when it has been tried in previous clinical trials, the challenge has been thrombocytopenia. And platelets, for some reason, are really very dependent on BCL-XL. The way we are thinking about overcoming that idea, I mean, it's a little bit, few steps further away from where we are right now, but the PROTACs, again, for reasons that are not completely understood, somehow are able to target nucleated cells way more than the cells lacking a nucleus. And the platelets somehow remain a little bit more resistant to PROTAC-dependent therapies. There are a couple of non-VHL directed BCL-XL PROTACs available now. We are trying to use those in our studies to get around that particular problem. So we'll see how this develops. The new agents, though, we are not worried about the off-target hits on the other BCL family. We are more worried about the on-target toxicity in other tissue. Oh, okay. So Vincent has a question, which I think I might have partly answered over here. So yes, Vincent. So the molecule that we are using, we reached out to AbbVie, and essentially, I think they don't have a BCL-XL program in solid tumors right now. I think their focus right now is in MDS, I believe. And so we originally wanted to start at least a phase 1 with them to see if this could be a viable strategy, at least in a subset of the tumors. That has not panned out. The inhibitor is great, though it is being used in human trials, but just not in the solid tumor space. But yeah, we are hoping that the work that we are doing will allow for some interest to develop in this idea and then make this a possibility in the future. That really is our ultimate goal. Yeah.
Fantastic. Okay, I'd like to invite our fourth speaker for this session, Dr. Neil Bhowmick from Cedars-Sinai Medical Center.
Thank you very much. All right, so I'll be talking on behalf of Peter Jones's lab, my laboratory, as well as Galing Liang. This was really a team effort that allowed me to stand in front of you today, so I'm very grateful. Oh, a little bit closer. Okay, thank you. But the real work was done by Hongtao Li, who's a postdoc, and he's started his own laboratory at this stage. So since I'm the last speaker right before lunch, I'm going to go ahead and give you the punchline right here on this slide. And you heard from Charles Swanton about SETD2, so I don't have to give you too much background, other than to say it's 15% of clear cell renal cancers have this mutation. And it's thought to be one of the drivers of the disease for these patients, especially metastatic progression and poorer outcome compared to other renal cancer patients without this mutation. Its job is to shepherd DNMT3B onto the gene body, as opposed to the promoter or enhancer in methylating intronic regions within the open reading frame, within the body. And so you're already familiar, whenever genes are turned on, oftentimes promoters and enhancers may be demethylated. However, these intronic regions within the gene body are actually hypermethylated, and SETD2 helps bring DNMT3B onto these regions. The reason we got interested in this is because in these intronic regions are these repeat sequences from ancient viruses that had infected humans and pre-humans long ago. And they have intercalated into these introns. When they get demethylated, these intronic sequences of these repeats, viral repeats, just pop up within the genome and are actually expressed through missplicing. And why is that important? Well, these viral sequences, when they're expressed, they form an event that allows for better immune response, because PD-1, PD-L1 markers that we see aren't necessarily a good indicator of PD-1, PD-L1 inhibitor activity. However, there are multiple papers now that have shown that ERVs, or endogenous retroviral transposon element expression, is actually a good indicator of response to immune therapy. So how do we act on this, on these patients, or in our case, mice? This is preclinical studies I'll be presenting today, is give 5-azacitidine or deoxy-5-azacitidine to cause demethylation and inhibition of DNMT3B and other DNMT proteins, as well as giving anti-PD-L1 or PD-1 inhibition on top of it. SETD2 has multiple roles. I'm only going to be talking about its role as a shepherd or as a helper for DNMT3B. So when SETD2 is knocked out from clear cell renal cancer cells, You see on the far left that there are more of these transposable elements that are expressed 5 days after treatment of 5-azacitidine has stopped. So this is long-lasting only in those cells that have a loss of SETD2, okay? So once you stop giving these demethylating agents, it remethylates and the virus sequence stop. However, in those somatic cells, those tumor cells that have the SETD2 loss of function mutation, these viral transposomal elements are still expressed. And in the middle section there, you see that we showed the mechanism of splicing that allows these intronic regions that are normally spliced out are actually expressed within the protein itself, and it results in elevated interferon expression that's shown on the far right there. So this is a summary of what I was just explaining, but on the far right is actually expression all the way 23 days after a DNMT inhibitor has administration was stopped. So the expression of these transposable elements are expressed way out long after DNA demethylating agents are given. Now, why do we have to give the demethylating agents in the first place? Well, because even though the SETD2 is lost, DNMT3 can find its way. Not very efficiently, obviously, but it can find its way. And so that's why you have to give the 5-azacitidine, or deoxycytidine. So to the mouse studies, we see that only in those cells, on those tumors that have a loss of SETD2, and this on the right here, and not those with intact SETD2, they actually have a response of the combination of PD-L1 inhibition and demethylating agent. Any one of these alone doesn't have that effect. You see more necrosis in the H&E. You can't see this very well in the dark field, but there is more recruitment of cytotoxic T cells in these. And again, sorry you can't see this, but this was mass cytometry to look a little bit deeper in the what cell types were actually being recruited to the tumor. And we see SETD2, I mentioned right at the beginning that they're more aggressive tumors, and these are actually more proliferative. This is Ki67 staining here. But they're more proliferative, more macrophage are being recruited, as are, of course, our T cells. Now, while we were doing this, another clinical trial was started and has just recently been published a couple months ago, using in fact demethylating agent along with PD-1 inhibitor. And this study was a phase 1 study. They didn't select for SETD2 specific patients because they weren't aware of the study, but we would really like to look at their data because unfortunately they did not see, they saw very good safety, but they did not see a positive effect that we're showing here with the mice. And with that, I'll stop and acknowledge the people in each of our 3 labs and our research support, so thank you. No, okay. So SETD2 has many functions, and I only talked about its role in recruiting DNMT3B to the intronic regions. And your question was, could you repeat one more time? So if you were to develop a drug against SETD2. Oh, No, we wouldn't, because the SETD2 is lost, but in 15%. But if you're speaking of the rest of the 85% of people, so there would be a problem in that. So you get inherent tumor selectivity with DNMT3B inhibitors like 5-azacitidine, because that somatic mutation is only in the tumor cell, and so you're not getting the toxicity. So I would be reticent in trying to make something against SETD2 itself, because it's ubiquitous in all your cells. Yeah, good question.
Very interesting work, thank you. Your theory presumes that SETD2 is crucial in this pathway of unmasking these viral particles that eventually act as immunogenic. Can you convince me that the SETD2 is truly necessary? Because at the end of the day, all these viral particles that are methylated are still incorporated in all these intronic regions that you have covered. And why not just use it for all comers, as the trial that you have just discussed? Because at the end of the day, demethylating these viral particles that eventually are expressed will still provide enough of immunogenic load and immunogenic stimulus to make it responsive to PD-1, PD-L1.
Yeah, so I think you sort of answered your question. So the trial showed that these patients that were on deoxycytidine, they did not have a positive response when combined with the PD-1 inhibitor, because they weren't being selective for the SETD2 deficient patients.
So again, what is it about SETD2 that really increases the sensitivity? And please forgive me if I missed this point, but to me it looked like it was the exposure and demethylating these viral particles that are supposed to come out.
Right,
what is the
right.
SETD2 doing that is so different from not SETD2?
Sure, sure. So a very important point that I glossed over was this splicing event that's described in detail in the paper, but just expressing these viral sequences, these viral repeats, is not sufficient. It actually needs to be expressed in a protein for the body to think that they have a foreign body that's there, to have an immune response, to have an interferon response to recruit the T cells. And so if you don't have SETD2, you don't have this aberrant splicing of the intronic sequences.
Thank you.
Yeah, thank you for allowing me to clarify.
Very nice work. Just a question, I don't know how much detail you've looked at this, but SETD2 obviously is a known driver, but also often a very subclonal event, or at least associated with polyclonality in a lot of the tumors. And have you looked, or is there an opportunity to look at in patients that have, for example, multiple regions that have been analyzed, do you see in the areas of SETD2 deficiency a more robust immune response that might be consistent with your hypothesis?
Yeah, I wish I could answer that. However, I would, because I'm here, I can make a guess, right? I mean, if there are subclones within a very heterogeneous tumor that has some SETD2 loss, you would have the recruitment of T cells in the region because of the interferon that's there, and hopefully the checkpoint inhibitor can turn it on. And since checkpoint inhibitors are quite promiscuous and can be non-specific, we would hope that some of the non-SETD2 mutant cells would get killed as well. But it's a total guess.
This exact data slide, the model that you're using, can you tell us a little bit about what context this experiment was done, and especially that same Okay. So basically the question is, what is the model in which this particular question is being addressed, and is it a surprise that when you're inactivating SETD2 in the untreated arm, there is no actual benefit in terms of tumor growth? It's not acting as a tumor suppressive event in this context.
You're asking if SETD2 is acting as a tumor suppressor?
That would be typically what our expectation would be from the 3P loss in at least the clear cell context. Yeah.
Right. We did not see it as a tumor suppressor at all. I see where you're going with this. So part of it is that this is normalized on the y-axis there for tumor size. And so where the blue, that means the wild type tumors are all normalized to the saline, whereas the red, the knockout, are all normalized to the saline. So I'm not showing the actual tumor size. The tumor size, you have a very good point. The raw data tumor size is actually significantly larger in the SETD2 loss tumors, as would be suggestive from, well, you can't see it, but did it come up? Yeah, there's significantly more Ki-67 in the SETD2 loss tumors, yeah. Yeah, it could be a little misleading.
Okay, very good.
Thank you.
Great. Thank you, everybody. I have one quick announcement. Everybody take a look at this number if you can. There it is. That's the number of grants that have been awarded to the recipients in this room. So I think we have a round of applause for this crowd. Just two quick things. One, pull out your calendar at lunchtime and mark down July 17 and 18, 2025. We're going to have another event here. KCRS25 will be here next year. And then secondly, or thirdly, if you have on your badge this lovely little aquamarine advocate on your badge, you're free to leave and go to lunch first since you were an advocate for kidney cancer. The rest of you have to stay behind until you get your advocate badge. So, okay, folks, time for lunch. Meet back here in about an hour. Thanks a lot.